Hello students. So in last video we saw how glucose is utilized through glycolysis. Now in this video we'll learn about the breakdown of monosaccharides other than glucose. And some of the common ones are galactose, mannose and fructose. Now do you see any similarities between glucose, galactose and mannose? If you want, you can pause the video and take a look at all the structures in the detail. Now, if you look carefully, you'll see that all of them have the same chemical groups, but their orientation is somewhat different. The glucose, galactose, and mannose are called epimers. Epimers means compound that have same chemical groups, but the arrangement of these groups is different around one or more chiral carbons. Now, what are chiral carbons? So, chiral carbon is a carbon which has four different chemical groups attached to it. And when there are four different groups attached to it, the orientation of the group matters. You can see in these structures that for the glucose on the third carbon, that is third when we count starting from the right of the oxygen. So somewhere here, this OH is on the upper side, and this on the fourth carbon, the OH is on the downside. Now, when you look at galactose, the OH on the fourth carbon is on the upper side. So this means that glucose and galactose are epimers on the fourth carbon. In the same way, if you look at glucose and mannose, the hydroxyl group on the second carbon is on the downside, and in case of mannose, it is on the opposite side. Now, fructose has a different structure than all three, so it is not an epimer. Now, we'll look at the breakdown of all these three sugars apart from glucose, like how they are utilized, whether they have separate pathway or they feed through glycolysis. So first we'll start with fructose. So the fructose is converted to fructose 1-phosphate by use of ATP by enzyme fructokinase. Now all of these conversions will happen in the liver in case of mammals. Now this fructose 1-phosphate is broken down by fructose 1-phosphate aldolase into two compounds, that is dihydroxyacetone phosphate and glyceraldehyde. Now glyceraldehyde is further phosphorylated by enzyme triose kinase using ATP to give glyceraldehyde 3-phosphate. Now if you remember from yesterday's video, both of these compounds, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, are intermediates in glycolysis pathway and they can directly enter glycolysis. Now on to the second monosaccharide, that is galactose. So galactose is first converted to galactose 1-phosphate by enzyme galactokinase, again using ATP. Now galactose 1-phosphate is converted to UDP galactose by enzyme UDP glucose galactose 1-phosphate uridylyl transferase. So what happens is a UDP glucose is added to the galactose 1-phosphate, and glucose 1-phosphate leaves the enzyme and the UDP group is transferred to galactose. Now this glucose 1-phosphate is converted to glucose 6-phosphate by the enzyme phosphoglucomutase and it enters glycolysis as glucose 6-phosphate. Now what happens to UDP galactose? So UDP galactose is converted to UDP glucose, and as we saw at the start of this video, these are epimers on the fourth carbon. So the enzyme UDP glucose 4 epimerase converts UDP galactose to UDP glucose, and this UDP glucose can be again used in the reaction, the second reaction. So this is where the epimerization takes place. Now we saw that there are three enzymes that are being used in the processing of galactose. That is galactokinase, the uridylyl transferase, and epimerase. Now, what happens if, because of some mutation, these enzymes are not functional? It gives rise to galactosemia. So, what happens in the case of kinase enzyme deficiency is high galactose in blood and urine, and it can lead to cataracts in infants. Now, out of all these three enzyme deficiencies, the deficiency of transferase is the most severe. It leads to poor growth in the children, speech abnormality, mental deficiency, liver damage, and it might even be fatal. And these symptoms will be there even if galactose is removed from the diet. Now, main source of galactose in the diet will be milk. The disaccharide lactose, which is mainly present in milk, is composed of glucose and galactose. Now, for the third deficiency, that is epimerase deficiency. In that case, the symptoms are similar to that of the transferase deficiency, but the severity of the symptoms is lesser. Now, moving on to the third monosaccharide, that is mannose. So, mannose is phosphorylated to give rise to mannose 6 phosphate by enzyme hexokinase. As we saw in the glycolysis video, that hexokinase is also the one which phosphorylates glucose. Now, this mannose 6 phosphate is directly converted to fructose 6 phosphate by the enzyme phosphomannose isomerase. And since fructose 6 phosphate is intermediate in the glycolysis pathway, so it can directly enter glycolysis. So what we have learned from this video is that all of the monosaccharides are processed through the same pathway, that is glycolysis, and they are converted into one or the other intermediates. Now, in the next video, we'll learn about the synthesis of glucose when cells are deficient in glucose, and that process is called gluconeogenesis. Thank you for watching.